In this chapter, we are going to talk about lipids. Now, the group of compounds called lipids is made up of substances with widely different compositions and structures. Unlike carbohydrates, which we just looked at, which are defined in terms of structure, lipids are defined in terms of a physical property, solubility. So lipids are defined by their solubility. Lipids are biological molecules that are insoluble in water. But soluble in non-polar solvents, right? Lipids are generally waxy, greasy, or oily compounds found in plants and animals. Lipids tend to repel water, which is a useful characteristic for protective wax coatings found on some plants. Fats and oils are energy rich and have relatively low densities. And so here's some, we talk about olive oil or coconut oil or avocado oil or the fats that are found in these foods. These all properties also account for their use as storage forms of energy in plants and animals. And so we're going to talk in this particular chapter about saturated fats versus unsaturated fatty acids. And other lipids are used as structural components, specifically in the formation of cellular membranes. And we have a cellular membrane here. And so we have the cell here. And I just want to remind you, the cell has a nucleus, and then there's cytoplasm, and then there's a cell membrane that kind of encapsulates it all. And the idea is that the cell membrane will separate things from the outside of the cell and the inside of the cell, but things need to pass through every so often. And so the cell membrane is a lipid bilayer. Bi just means two. There are two layers of it, and these are made up of lipids. And so it forms a really critical function for the cell where it only allows certain things to pass through using some protein channels, which we're going to talk about later. We can see some carbohydrate side chains that are here that help with molecule recognition. And then the yellow here is cholesterol, which is another type of lipid. So this specifically is called our phospholipid bilayer, which we'll talk about more as we get on into the chapter. But this is a really nice overview. So without further ado, we're going to jump in and talk about some of the classes of lipids. Now, lipids are grouped into two main classes. The first class is saponifiable lipids. Okay. And then the second class is non-saponifiable lipids. So those are our two classes. Now, saponification refers to that process where esters are hydrolyzed under basic conditions. So when we talk about saponification, just as a review, this is just the process where esters are hydrolyzed under basic conditions. And we talk about this because, this is one of the ways that we make soaps, right? Is from these fatty acids. So things that can undergo or are saponifiable lipids are things like triglycerides, waxes, phospholipids, and sphingolipids. Right? And all of these are esters. And so all of these can go through saponification. They are later divided into simple and complex. So we have the lipids here. Here are two categories. There are simple saponifiable lipids, which are waxes and triglycerides. And then we also have complex saponifiable liquids, which are things like phosphoglycerides and sphingolipids. Now, non-saponifiable lipids are not esters and cannot be hydrolyzed. Right? And so the steroids and prostaglandins are two categories are two types of lipids that belong to this category. I want to go back and talk about the two categories here and how they differ. When we talk about these simple saponifiable liquids, these contain just two types of components. Generally a fatty acid and an alcohol. Then complex lipids, which are over here, right, consist of more than two types of components. And so in general, they tend to be a fatty acid, an alcohol, and something else. All right, so a fatty acid, an alcohol, and something else. When we talk about lipids, we also want to or need to discuss the functions of lipids. Fatty acids are lipids generally repel water. It's one of the characteristics for particularly this is the protective wax coating found on some plants. It's also found on bird feathers. They're also energy-rich compounds with low density, so they're used as storage forms of energy in plants and animals. And then they're also used as structural components, especially in the formation of that cell membrane. Now, at this stage, I want to zoom in and talk about fatty acids. Fatty acids are a component of both saponifiable and non-saponifiable lipids, and so we really want to define that before we go on. So as we talk about fatty acids, these are really the fundamental building blocks of lipids. Right? Fatty acids are just long chain carboxylic acids. And so we see here an example of that where we have a long hydrocarbon chain and then we have a carboxylic acid here. This is a nonpolar region, and so it's called a nonpolar hydrophobic tail, and that means that it's water insoluble. This is the polar region, and that's hydrophilic, and it's water soluble. And so this a lot of times is called a polar head. And so in a cartoon image of these fatty acids, which we'll see represented through different types of lipids, you'll see here that we have the polar head here, and that's the carboxylic acid group. And then we have the nonpolar tail here as well. So let's put a little bit of that into words. The long nonpolar tails are responsible for most of the fatty or oily characteristics of fat, right? So then we have the carboxyl group, or the polar heads, of fatty acids are very hydrophilic. And exist at physiological pH as a carboxylate anion, which is COO minus. Okay, and I know that it shows you here the COOH, but this minus is important because that means in a living system, it's going to have this negative charge, and then that will form interactions between that and the water molecules. Now, what happens here is with particularly fatty acids is they can form micelles in aqueous solutions. Now, a micelle is just a spherical cluster of these fatty acid molecules. So a micelle is a spherical cluster of fatty acid molecules, right? Where the polar chains are on the surface and the nonpolar chains are located in the interior and are held together by weak intermolecular forces, which I'm going to abbreviate as IMF, right, which are dispersion forces. So when we look at this, kind of the cartoon version here is we've got those non-polar tails on the inside, and then we have the polar heads on the outside. And then these can come along and interact then with water molecules. And they're going to react with the hydrogen of the water molecule because that's partially positive, and then that oxygen is partially negative. And so it's the partial positives that interact here with these negatives because positive and negatives attract each other. Now, when we look at this, these can contain hundreds or even thousands of fatty acid molecules. So this image is somewhat misleading, but it still is helpful. Micelle formation and structure are important in a lot of biological functions, like the transport of insoluble lipids in the blood. So let's see. These micelles are important for biological functions, such as the transport of insoluble lipids in the blood. Now, another place that we see micelle formation that's really important is in soaps. So how soaps work is that that they form a micelle here, and the interior here is nonpolar. And so what happens then is that dirt is also nonpolar, and oil and things like that, go into the interior of the micelle. And so when you like, for example, have dirt or oil on your hands and you go to wash that and you lather up the soap, that forms the micelles, and the dirt and oil go inside the micelle. And then the polar heads here interact with the water and they get washed off your skin. And when they get washed off your skin, dirt and oil also get washed off with it. And so that's how soaps work. So when we talk about how soaps work, greasy dirt is not soluble in water. And so when the skin or clothing, when skin or clothing has a like greasy dirt stain and it's washed with soapy water, the stain or the greasy dirt is attracted to the non-polar hydrocarbon tails. So the greasy dirt is attracted to the non-polar hydrocarbon tails of the soap. Right? And then those are dissolved in the interior of the micelle that's formed by those soap molecules. Because that surface of the micelle is covered with polar head groups, the entire micelle is soluble in water and is washed down the drain. And so that's what removes the dirt. Now, when we talk about this particular molecule here, it is referred to as an amphipathic molecule, which just means it has a polar and non-polar sections. Right? And so these amphipathic compounds, like soaps, are referred to as emulsifiers. And emulsifiers just allow polar and non-polar compounds to be suspended in the same mixture. And so this is one of the applications of micelles that we observe. Now, let's talk a little bit more about fatty acids. Fatty acids are usually straight chain carboxylic acids with no branching. Now I'm going to abbreviate carboxylic acid as Ca. They generally range between 10 to 20 carbons. And they usually have an even number of carbons. This has to do with the way they're synthesized. Right? And what we also know about these is they can be saturated, which in this case means they contain no carbon-carbon bonds, or unsaturated, which means they contain one or more carbon-carbon bonds, usually in the cis configuration. Apart from the carboxyl group and double bonds, there are usually no other functional groups present. I want to remind you with the cis configuration, this means the hydrogens are on the same side of the double bond, and then the long chains are on these sides here, and it tends to form a U shape that looks like that. Let's look at some important fatty acids. So here's a table of fatty acids. I want you to notice that it's broken up into saturated fatty acids. These have no double bonds. Here are their melting points, and what we can see is as the number of carbons increase here, the melting point also increases in saturated fatty acids. And here are some places that we see saturated fatty acids. We can have monounsaturated fatty acids. Monounsaturated fatty acids just mean they have one double bond. And then we can have polyunsaturated fatty acids, which means that they have more than one double bond. Now, I want us to talk about unsaturated fatty acids for a moment, but before that, I do want to put some words to, right? In saturated fatty acids, as the number of carbons increase, the melting point also increases. And that's because we end up with more intermolecular forces. When we go over here and talk about unsaturated fatty acids, we have now a double bond. So if you look here, this is a saturated fatty acid. It has a long nonpolar tail. It's all single bonds. This is an unsaturated fatty acid, and we have a double bond here, and it forms, right, kind of this U shape here. And then linoleic acid has two double bonds, and the U shape is even more pronounced here. So a cis configuration creates a kink or bend in the fatty acid chain. And what that does is it prevents the chains from packing closely together. And so what that does is it leads to weaker intermolecular forces and a lower melting point. And so if we look at stearic acid right here, it's got 18 carbons and its melting point is 71. Oleic acid, which is right here, also has 18 carbons and it's got a double bond, and so now its melting point is 13. So the fact that these molecules can't get really close to each other and have those intermolecular forces, that really does decrease that melting point quite a bit. And then when we have 18 carbons and two double bonds, it decreases even more to -5 here. Most fatty acids are liquids at room temperature. I'm going to abbreviate room temperature with RT. And then, right, the melting point decreases as the number of carbon double bonds increase. And what we see is that the fluidity of biological membranes can be explained by the length of the chain and the presence of double bonds. And we're going to discuss that more in section 18.8 when we get to talking about those membranes. But I wanted to just kind of give you a preview of that. Now, the body can synthesize all except 2 fatty acids it needs. And those 2 are linoleic acid here and linolenic acid here. And these are polyunsaturated fatty acids that contain 18 carbon atoms. And what you see here is that's that 18 in that nomenclature there. And then if you see a colon here and then another number, what that means is that it has 2 double bonds. So the 18 here is the number of carbon atoms, and then there's a colon there, and then there's another 2, and that tells us the number of double bonds. And then you're going to see either this or sometimes a little omega here, like omega 6 and omega 3. And so when we talk about an omega 3 fatty acid, all that means is that we come here and we from the side of the molecule that doesn't have the carboxylate, we've got 1, 2, 3, and on the 3rd carbon there's a double bond. So from the methyl side of the carboxylic acid on the 3rd carbon is a double bond. And then an omega 6 fatty acid has a double bond on the 6th carbon from the methyl. Because these 2 fatty acids, they're essential fatty acids because they are not synthesized in the body, and that means we must obtain them as part of our diet, right? And so that's why they're called essential fatty acids. It's essential that we obtain them as part of our diet. Both of these are pretty widely found in plant and fish oils, as well as some nuts. In the body, both of these acids are used to produce hormone-like substances that regulate a wide range of functions and characteristics, including blood pressure and blood clotting and blood lipid levels, immune response, and the inflammation response to injury and infection. So these are absolutely critical. Omega-3 fatty acids, which this is an example of, have been the topic of interest in medicine since about the mid-1980s. In 1985, the researchers reported the results of a study involving people in Greenland, and basically those individuals have a really low death rate from heart disease, even though their diet is really high in fat. And so studies led the researchers to conclude that the abundance of fish in the diet of those individuals was involved in some way. And so continuing studies led to a possible involvement of omega-3 fatty acids in the oil of the fish, which may decrease the serum cholesterol and triglyceride levels. And then a recent 5-year study on the benefits of omega-3 fatty acids involving about 18,000 patients with unhealthy cholesterol levels showed that at the end of the study, patients who took omega-3 fatty acids had much better cardiovascular function, and the non-fatal coronary events like heart attack were significantly reduced. So we're finding that these two fatty acids are really critical for good health. So this ends our discussion of fatty acids. As always, if you have any questions, please feel free to stop by my office.